Hello students, welcome to our preclinical pharmacology channel. Today we are going to discuss some introductory part about the reproductive toxicology. This is very important topic as pharmacology and toxicological screening methods are concerned, and that is there with the syllabus of M.Pharm pharmacology. So let's start with the introduction part. What is reproductive toxicity? So reproductive toxicity is a study of occurrence, means appearance, then causes, manifestations, and sequential adverse effect of exogenous agents on reproduction. This is very important that the effect of exogenous agents on reproduction, which always occurs and you observed, are nothing but the study of all these things are nothing but the reproductive toxicity. Now, what is developmental toxicity? It is actually related to the abnormal structure or functional development following exposure of pregnant or lactating females. So, again, it is the same, but when the exposure to exogenous agents will causes the abnormal structure or functional development in the fetus during the pregnancy and lactation female, the study of all these things are under the developmental toxicity. Now, this reproductive toxicity is the subject which dealing with the causes, mechanism, effects, and prevention of disturbances throughout the reproductive cycle, including fertility induced by the chemicals. So, this is another way by which you can define the reproductive toxicity. Then the reproductive toxicity is mainly occurs because of the exposure of human reproduction to xenobiotics, such as drugs. Various drugs will affect on the reproductive system. Then substances of abuse like morphine, codeine, all these things. Then industrial chemicals. Various industry, they releases the chemicals which are really toxic to the human body. Then pesticides like DDT and all these things. Airborne contaminants. Then trace metals. These metals like cadmium, lead, and arsenic, which are there in the nature in very trace level. And food additives and lifestyle. So apart from all these things, lifestyle plays a very important role in reproductive toxicity. Then the causes of this infertility, as we have seen here, that these are the different causes which leads to the human reproduction problem in human reproduction. But still, there are one third male and one third female, and one third between the couple. The causes are unknown and they are remain unknown, and that's why many a time it happens that everything is managed, but still the pregnancy is not occurs. So this reproductive hazards means the what problem arises once the reproductive toxicity occurs. These are produced in the mothers and father as well as the unborn and the newborn child. So this includes loss of libido, then sterility, mutagenesis, teratogenesis, abortion, fetal death, perinatal death, and delayed toxicity. So these are the mainly major effect which occurs on reproduction by the exposure to various these hazard materials. Now, if you see what are the components of male reproductive system, so this figure indicate that these are the various components of male reproductive system like testes, epididymis, then vas deferens, prostate, glandular vesiculosa, then glandular bulbourethralis, then urinary bladder, urethra. So all these near about 15 components are there which plays a major role in male reproductive system, and this allows the male to produce sperm cells which gets the female pregnant. So Here, this system is involved in the spermatogenesis, and these sperm cells are responsible for uh, producing the pregnancy in the female. Likewise, female reproductive cell, they are having the various components similar to the male. If you observe here, the main components are the ovary, then oviduct, uterus, cervix, vagina, urethra, then different parts are there, that is labia minora, labia majora, and then colon, then rectum and anus. So 20 different components or parts are involved in the female reproduction, and it has been observed that the female reproductive cycle is quite complicated as compared to the male, and this system is involved that the female to make the eggs and to get pregnant to produce babies with the male. So by in that way, the male reproductive organs and female reproductive organs both are equally important and involved in the development of the newborn babies. Now, what are the neuroendocrine uh, system which are involved in the reproductive toxicology? So neuroendocrine targets. So whenever these xenobiotics are affected on the reproductive system, so what are the mechanism by which it produces the reproductive problems? And that includes like environmental toxicants, and these environmental toxicant can disturb the reproductive function mainly by the three different mechanism. First is the endocrine system, that is they alter the endocrine system. They are called as endocrine disruptors, and they mainly disturbs the hormonal system. Another is the altering gene expression, and that is pertaining to spermatogenesis as well as the steroidogenesis. And third is the exerting epigenetic effects, and which can be result in abnormality in the reproductive system of male offspring up to the fourth generation following in utero exposure. Next is the generation of oxidative stress. Nowadays, oxidative stress plays a very important role, and change in the lifestyles are the one of the factor for this oxidative stress. And many metal ions and environmental toxicants, they produces the oxidative stress. So it has been observed that this oxidative stress can be seen in up to 80% of clinically proven infertility man, and exposure to environmental toxicants is a major factor contributing to such increase. So this plays a, and nowadays many research is going on related to the role of oxidative stress in various disorders and diseases. Now, endocrine disruptors, which is related to hormonal irregulation, may exert their course of action by acting on either as agonist or antagonist at the receptor site, thereby minimizing or blocking the cellular response in such endocrine gland as the adrenal, thyroid, and ovary. So these endocrine disruptors, they act on the receptors which are present at different sites, and they blocks the cellular responses. And next is the altering any of all these system can lead to impaired neurological development depending on the timing and duration of exposure of the offspring in the pregnant woman. So this is very important line, that though these are alters the neurological development, but still it is depending on the time of exposure and duration of exposure of the offspring in the pregnant woman. Then immune toxicant. So some toxicants are there which affect the immune system. So immune toxicants, they disturb the cytokine secretion by macrophages and activated T cells, thereby preventing normal activation of other T and beta cells. So thus, these chemicals may disturb cytokine mediated communication between immune cell, impairing normal immune responses that may be fundamental to reproduction. So these are the neuroendocrine targets of the reproductive toxicity. Now here, this hypothalamic pituitary gonadal axis plays a very important role or we can say that it plays a central role in reproductive toxicology. So in your previous classes, you have studied this endocrine system. So just for understanding, if you can observe here that this hypothalamus, it increases the production of the gonadotropin releasing hormone, that is GnRH. And this gonadotropin releasing hormone, they shows the effect on the pituitary, anterior pituitary, thereby releases the luteinizing hormone and follicle stimulating hormone. So these luteinizing and follicle stimulating hormone, they have the various effects on testes as well as the ovaries, and thereby they produces testosterone and estrogen from the ovaries, and they have the different effect on the reproductive organs. Like estrogen, if estrogen is released from the ovary, it causes folliculogenesis. Then it will also responsible for female secondary sex characters like breast development and maturation, hips broaden, and pubic hair growth. Likewise, if testosterone are releases, they are responsible for spermatogenesis, and apart from that, the male secondary sex characters are developed because of this testosterone, including the penis and scrotum growth. Then facial hair grows, then larynx elongates, lowering voice, shouters broaden, then body, armpit and pubic hair growth, and musculature increases and body weight. So these are the uh, hormones in male as well as the female, which are responsible for secondary characters and the main process of reproduction like folliculogenesis and spermatogenesis. They also having the some feedback mechanism. Now, what are the endocrine disturbers and female reproduction? Means when endocrine disturbers affect on the female reproduction, what are the outcomes? Here we have discussed with some species like marine mollusk. There is a morphological changes in reproductive organs. If it occurs in the fish, it reduces fertility and hermaphroditism. If it occurs in the birds, then abnormal clutch size, female-female pairing, embryonic deformities. In reptiles, it produces abnormal sexual development, and in women, it increases risk of breast cancer, early puberty, altered menses, decreased fertility, increased miscarriage rate, and early menopause. Similarly, if this endocrine disturber effect on the male reproduction, it also produces several different types of the alteration. Suppose the male reproductions are exposed to the organochemicals and pesticides like DBCP, DDT, and digoxin. These are given. Or heavy metals like lead, mercury, cadmium. It produces decreased fertility, decreased libido, embryo fetal loss, birth defect, cancer, estrogen effect, poor semen quality. So these are the different effects on the male reproductive system after exposure to this environmental, then biological and pharmacological agents. So here the details are given. Then what is the mammalian reproductive cycle? This is the general picture of the mammalian reproductive cycle. So it start with the sexual maturation, then come to the release of gametes, then fertilization, transport of zygote, implantation, embryogenesis, fetogenesis, birth, then fetal development, development and growth, and again fetal. So this embryogenesis can also involved in the maturation and production of the gametes, which leads to release of the gametes. So this reproductive toxicology, this covers a wide spectrum of toxic effect at all the stages of reproductive cycle, starting with the female and male fertility. From here, it will start, and prenatal and postnatal development, and culminating in late manifestation that can only be detected in the next generation. So it is a complex phenomena, and in this toxicant will affect on any of the stage of this reproductive cycle. So Based on this, in 1966, the US FDA was the first regulatory authority which issues a series of guidelines for evaluating the novel pharmaceuticals for their effects on reproduction function, and develop this guideline describe three studies. And these three studies including the segment 1 study, which is the fertility, segment 2, that is embryo toxicity or teratogenicity, and segment 3, that is prenatal and postnatal toxicity. So in the same reproductive cycle, if you observe what is the segment 1 fertility, so here fertility it is given in the figure that segment 1, it involves production and release of gametes, fertilization, transport of zygote, implantation. That is actually the mating process. Then segment 2, it is also called as the teratogenicity, and it involves actually the gestation period like embryogenesis and fetal development. So this is segment 2 of this study. And segment 3, that is prenatal and the postnatal toxicity. Here you can observe prenatal and postnatal toxicity. It involves the parturition, lactation, postnatal development, development and growth. So this part involves the segment 3, and this part involves segment 2, and this involves segment 1. So as per the syllabus, how to detect, how to study the segment 1, segment 2 and segment 3. For that we will going to post a separate video for all the three different segment study. These are again another picture which gives you the idea about the segment 1, segment 2 and segment 3 study. What are the different steps are there and where this will affects. Then for carried out the reproductive toxicity, there are certain essential features which one has to understand. That is, adverse effect may only occurs in the next generation or offspring. So many a time it's happened that if the toxicities occurs, but the adverse effect you can notice in next generation or the offspring. Then adverse effect on the vital function of all organs may also indirectly induce adverse effect on reproduction itself. Means if the vital organ is affected, then you can observe the effect indirectly on the reproduction itself. Then, if you want to assess the potential of chemicals to interfere with reproduction, so test method in experimental animal must cover the following essential steps. First step is that growth and maturation of the sperm and oocytes are important. Then fertilizations is important like fusion of oocytes and sperms. Then normal cleavage division, implantation, intrauterine development, birth and postnatal development throughout the period of lactation is also equally important. Then normal development of the offspring to fertile adult animals which are able to produce a second generation. So these are the some essential criteria, essential steps which are required if you want to check the potential of chemicals on the reproductive toxicity. Then next is there are certain guidelines for study the reproductive toxicity. These are the guidelines. ICH guidelines are there which are used for the segmental study like segment 1, segment 2 and segment 3. Apart from that, OECD guideline is there, that is for industrial and organochemicals. This is for the drugs. Industrial and organochemicals, you have various guidelines like teratology study TG414, one generation study TG415, two generation study TG416, reproduction and developmental screening, then combined repeated dose and developmental screening, developmental neurotoxicity study, uterotropic assay, osteogenic properties. So all these OECD guidelines are also available on the website. Then, what are the species required for evaluating the reproductive endpoint? So, if you are doing the study in case of the male and is well in different species like mouse, rat, rabbit, pig, and monkey. So, if you are using the mouse and it is male, then you have to evaluate this much parameter like spermatogenesis, testicular pathogenesis, epididymal sperm, fertility, in vitro fertilization. If the mouse is there but it is a female, then you go with the embryogenesis, ovarian pathogenesis, estrus cycle, endometrium. In case of the rat, you can go with this much of the parameter in case of male, in case of female. In rabbit, these are the parameter. In case of female rabbit, you can evaluate this parameter. In case of pig and monkey, if male is there, there is no specific parameter is reported. But in case of the female, you can go with the following parameter. So this is all about the general basic introduction of reproductive toxicology. The details about the male and female reproductive toxicology and the different segments we will going to post as soon as possible. So if you like this video, then circulate in your friends, and also like you give the comments what you are expected from this channel. I am always welcome to solve the problem. Thank you once again for your patience listening. Thank you.